Welcome to the Genome Sciences Annual Symposium, co-sponsored this year by Computer Science and Genome Sciences and the Fred Hutch. We're very happy to have you all here. And it's my great pleasure to introduce our next speaker, David Baker, who I feel like probably needs very little in the way of introduction because you've probably read bios about him in the newspaper this week after the Nobel Prize announcement last week. I'll just briefly say I was looking at all of his bios and I thought it was cool to some of the numbers: 640 peer-reviewed publications, 100 patents, 21 biotechnology companies that he co-founded, and 90 of his trainees have gone on to faculty positions elsewhere. So in addition to, of course, all the fantastic science, I've known David for 25 years and it's been great fun working with him over those years. And I look forward to hearing your talk, David.
All right, thanks, Bill. It's been great fun to be here and work with Bill and many of the people here. And right, so we've been working on protein design, and I'll tell you a little bit about the methods we've been developing and then what we've been doing with them. We started dividing the problem up into several steps. So let me back up a little bit. So why do we care about proteins? You know, proteins carry out all the important functions in our bodies and in essentially all living organisms, and they solve all the problems that came about during biological evolution, but they don't solve new problems that we face today. So the idea of protein design is to make new proteins that have new functions, both to learn about biology and hopefully to make the world a better place. Okay. Let's see. So we divide the problem up into several steps. So first, given a new function you want to make, generate a protein backbone which has the right shape to carry out that function. And then once you have that new protein backbone, find a sequence which will fold to that backbone and have amino acids in the right place to have that function. And then finally, once you have that new sequence, predict the structure and see whether it's actually predicted to fold to the structure that you were aiming for. And so, and this we do with RosettaFold, the program we developed, AlphaFold developed by DeepMind, and there are a number of different increasingly accurate structure prediction methods to use for the last step. So how does the backbone generation part work? Well, it's really inspired by methods for generating images, so generative AI methods. And in fact, it's kind of what's kind of neat is the computer vision community keeps developing new exciting algorithms, and we pretty much look through, or I don't do it, my students look through kind of the latest in that literature with the things people are posting, and then we try them out on proteins. And I'll sort of illustrate how that works for diffusion. So in the image case, to train a diffusion model or other flow matching or whatever model, you start with a very large number of images that you take from the internet. You noise them to different extents, and then you train a network to remove the noise. And obviously, it's sort of a different feature, different noise scales, different things are being learned. Like, you start at high resolution, start learning about edges and contrast. At low resolution, features more probably like homogeneity, and lack of really high frequency variation between pixels. The neat thing is once you've trained a model that, given a noised image, and importantly, the amount of noise that was added, can predict the original image, and obviously it gets increasingly hard as you go further away from the actual image. Once you have such a method, you can start with completely random noise and progressively denoise it to generate something that looks like it could have been in your training set, but is actually completely new. And you can condition this process, as all of you know, on a text prompt, if you include text fragments during training. So you can ask it to generate a picture of a cat sitting on a table. Well, so we just copied this exactly for proteins. So we start with a a protein with every protein structure in the PDB. We noise each residue to we apply different amounts of noise, and then we train a network. It's actually a variant of our structure prediction network, Rosetta Fold, to predict the ground state, and or predict the original structure. So we take each protein structure in the PDB, we noise it to different extents, and then we train the network to remove the noise and regenerate the original structure. And once we've done this, we can start with a completely random distribution of residues and repeatedly denoise. And what we end up is something that looks like a new structure, but actually is completely new. And this is what you're seeing here is exactly just an animation of this. And so you see, we end up with something that, for those of you who know, know about proteins, looks like a protein, but starting from completely random noise. And this is just looping. So that's not. So what we did early on is once we had those backbones, we used protein MPNN to generate sequences that were predicted to fold those backbones, check that they were actually predicted by AlphaFold or Rosetta Fold to predict those, to go fold those backbones. And then we made synthetic genes encoding those proteins and put them in bacteria. So we have to make synthetic genes because these are brand new proteins. They have completely different amino acid sequences from anything in nature. So we take those synthetic genes, we put them into bacteria. The bacteria essentially become factories to make the protein. We get the protein out, and we determine whether it has the structure or function we wanted to make. Okay, so we found we could make all sorts of new protein structures that way, but that really wasn't very useful. So what I'm going to tell you about for the rest of my talk is making proteins that have functions. So here's the insulin receptor, and so we can condition the diffusion process by, during training, in some cases providing parts of a protein and keeping it fixed. And so the model learns what protein interactions look like. And so then during the denoising process, what we get out is a protein that really, hugs the contours of the surface very, very closely. And so this is just a denoising trajectory. It results in a backbone, and then we can again design a sequence for that backbone, make the protein in the lab, and see whether it binds. So I'll now show you several examples of this. Here we have the TNF receptor, and we start with the random noise, we denoise it, and we end up with these proteins that really hug the surface of the receptor. When we make them, they bind reasonably tightly to start with. If we take these designs and add a little bit of noise and remove the noise, we get down to very, very tight binding, 9 picomolar. It's actually interesting. This is the tightest monomeric binder to this target, despite it being really of central interest in the pharmaceutical industry for suppressing inflammation. And we can take these binders and optimize them for other members of the family just by repeating this partial diffusion. We can also condition on the type of structure we want to generate. So there's a lot of interest in the pharmaceutical industry in antibodies. And so during training, we can specify the fold of a protein. And so we can take a structure then and point to a region on the surface, and then carry out this denoising process and say in addition that it should be an antibody fold. And what's kind of neat is it's generating structures. These are for those of you who know about antibodies, they have these complementary defining regions called CDRs. And so this process is building up the whole antibody, including the CDRs, and positioning the dock. And when we solve structures of these proteins bound to their target, this is a comparison of the cryo-EM structure and the diffuse structure, and you can see they're very similar. So we can also target unstructured proteins. And here I'm showing you results on targeting peptides that form amyloid that been invoked in or implicated in neurodegenerative disease. So this is the beta peptide, and this is a portion of tau. And we get these proteins out that really kind of sandwich the peptide. And in vitro amyloid formation assays, where you take the peptide, if you put it in solution, it will form amyloid over time. That's what we're looking at here. If you add these binding proteins at stoichiometric concentrations, they completely suppress aggregation. So we're excited about applying this sort of methodology to neurodegenerative disease. KJWU has taken these methods or developed methods for quite generally designing binders to disordered proteins, which have typically been very hard to target. So this is just a list at the top of some of the proteins that she's made binders to. And here's a crystal structure of a binder to dynorphin, neuropeptide, and this is, again, comparison to her design model. And what's neat is that these binders are really quite selective. You can see that the sequences are pretty different of the targets, but they're all disordered. So you might expect that the proteins that bind to these disordered proteins would have considerable crosstalk. But here are these peptide sequences along this axis, and then here are binding proteins for those targets. And you can see they're really very highly specific. So we're excited about this for proteomics applications, among other things, because we can make binders to pretty short peptides. And you could imagine that if you had binders to a sufficient number of short peptides, you could come up with much faster ways of doing proteomic analyses. We found, working with Mike Macasa's group, that we can enrich for peptides prior to mass spec. And Gage has been working with Mike Gilb's group on newborn screening methods, where similarly you can take a blood sample, very rapidly enrich for a particular peptide or class of peptides to diagnose disease. And that's looking very promising. So I've described design of binding now, design of binding, and now I'm going to switch gears and talk about design of transmembrane protein sensors. So we've been working on designing, de novo designing membrane pores like this. These are beta barrels, and this has been a wonderful collaboration with my former postdoc, Anastasia Vorbieva, who's now in Belgium. And the reason this was sort of an interesting challenge to begin with. These have a hydrophobic exterior, so once we make them, we can add them to planar lipid bilayers, and they'll insert. And what we find is that as the pore gets bigger by increasing the number of strands, the conductance gets larger, and that's sort of neat. So we can make pores from scratch. So there's a lot of interesting applications of these. One is, Oxford nanopore sequencing relies on a naturally occurring pore, but now we can make them completely from scratch and really custom make them for sequencing, DNA sequencing. We're really particularly interested in protein sequencing because, again, we can change the width of the pore, the lining of the pore really at will. And it's been a really fun collaboration with my friend Jens Gundlach here. We've also been working on design of small molecule binding proteins, and this shows a design binder to cholic acid made by Lena Anna, postdoc in the lab. And Lena and Sagardip and a rotation student, Valentina, took this binder, her binder, and basically fused it to the top, broke it into parts, and fused the parts to the top of the pore here. And so this is that sort of this pore with the de novo beta barrel here with the fragmented cholic acid binder coming out. And this is what the current looks like in the absence of cholic acid. And when cholic acid is added, you get this dramatic change in profile. And here you can see complete shutoff of the receptor. So we're really interested in making sort of a general electronic nose from this because we can make quite a number of different nanopores. And again, like I said, we can change their lining, we can change what's above. And then you get a lot of rich information from these sort of traces. So it becomes kind of an interesting ML problem where you ultimately would like to be able to take a solution of molecules and deconvolute what you have in there based on the response of each of your different nanopores to what's in the solution. And this is the collaboration with Jens's group. So Alexei and Jinwei are designing nanopores that have the appropriate size to fit into pores that have been drilled in silicon nitride chips. what the goal here is to directly incorporate sort of the biological, sensing, the biological, the protein with whatever sensing capabilities it has with silicon. Here are some of the really cool structures they've made, and it's looking very promising right now, and being able to actually get proteins to insert into silicon and give robust responses. So I thought I would say a little bit about DNA binding proteins. And so the way we've been approaching DNA binder design is very similar. So here we have a protein that's diffusing in the presence of DNA. And so when and then once we get a backbone, we can design the backbone and then again make synthetic genes. Here are some examples of designs made using this method and sort of previous sort of physically based methods, which I think many of you heard me talk about over the years. So here is a designed protein, and here's the DNA. And you see the designed protein has these side chains which make very specific hydrogen bonds with these target bases. And so this is work led by Cameron Glasscock. And what he showed was that if this is now experimental binding data, where he changes the identity of this target site one base at a time. And what you can see is when, for example, this C is changed to any of the other three bases, the affinity is decreased considerably. And the redder the color, the greater the decrease in affinity. Here's another example here as well. I think I left it out, but I did leave it out. But we can take these DNA binding domains and now hook them up to, we can use them to either repress or activate transcription. And we're now trying to incorporate additional activities, for example, nuclease activities, to make really small custom targeted nucleases. So now I want to talk about the design of assemblies. There's a lot of excitement a year or two ago when my colleague Neil King took a two-component assembling nanoparticle that we had designed, put the COVID RBD, spike RBD on the surface, and found that this was a very potent, elicited very, very potent responses against the immune responses against the virus. And this has now actually been approved for use in humans. So it's kind of our first de novo design medicine. So we've been very interested in these sort of structures for both for vaccines, but also for delivery. Biologics delivery is a big challenge today. And one of the problems we faced was that if you have strict symmetry, so the environment of every protein subunit in the assembly is the same, then the largest structure you can build is an icosahedron, because that's the largest of the regular polyhedra. Now, to go larger, and that sort of confines the packaging size within if you want to do delivery. Now, viruses overcome this by making larger particles, and they do this in a couple different ways. In both cases, what basically happens is you have pentagons. If you think about a dodecahedron, it has 12 pentagons, and you can expand a dodecahedron by inserting hexagons between the pentagons. And you can do this in one of two ways. You can either use a second type of building block here, because obviously the environment of this protein here is different from the environment here, because this one surrounds a 6-fold ring and this one surrounds a 5-fold ring. So one option is to just add a new type of building block. Another option is to use the same type of building block, but program in flexibility so that it can forms pentagonal faces in some cases and hexagonal cases in others. So this is the first approach where we start. This is work by Sangmin Lee and Ryan Kibler. We start with this icosahedral structure, and which has these 20 trimers, each surrounding a pentameric interface. And we kind of pull it apart and insert a new type of building block, which now surrounds hexagons, as shown here. So here are the original pentagons, and here's the hexagon. And this is the cryo-EM structure of this complex. And just to give you a feeling of scale, this is AAV for comparison. And so we're excited about being able to make these new type of these large cages completely from scratch. This one has actually 4 different components, which are shown in the different colors here. And so it's a little bit complicated. So we then tried to make Sangmin and David then tried to make cages that would be similarly large. And here's a cryo-EM structure again compared to AAV, but just using a single type of subunit. And the basic idea here is that if you have a hexagonal, if you have pure hexagons, you get a flat sheet. If you have pure pentagons, you get a closed icosahedron. And if you go somewhere in between and the sort of the angle between the axes that you might imagine coming out here, if you go for intermediate curvature, then you can get a variety of structures, including this complex here, which now has, again, it's got both pentagons and hexagons, and it's again got considerable packaging efficiency. So now the challenges are we've been able to put targeting domains on these, so we can target them to cells. The real challenge we face is how to get endosomal escape, and so that's still tricky. The question is whether we need to engage with host machinery, or whether simply sort of having a sufficiently large pH-driven conformational change will be sufficient to get through the endosomal membrane. Okay, so everything I've talked about so far are proteins that have a single low energy state, and we've been working to make proteins that have multiple low energy states. And this is actually the original switches here were developed by Florian Praetorius and Phil Leong a couple years ago. And they basically sort of schematically look like this, where there's you have one conformation of a protein, it binds an effector, and then there's a conformational change. And so in the absence of effector, you get, if you position these properly, you get this sort of square-like structure. When the effector binds, you get this hinging motion. And so now you have 3 of these that surround that make this triangular structure. And so Arvind Pillai made a number of structures like this that are really cool. So here's one that's square-shaped, and when the effector is added, it forms this pentagonal structure. So in this case, the angles open out, so you get a pentagon instead of a tetramer, and you can kind of see the square going to a pentagon here. And if you count the number of subunits in this particle, in these assemblies using mass spectrometry, you see they go from 4 to 5. And here's another example of one that goes from 3 to 4. So there's sort of some classic cases when you learn biology and biochemistry of things like hemoglobin, which have 4 subunits, and then an effector comes in, oxygen in that case, where there's a concerted conformational change. So Arvind wanted to design systems like that. So here you have, there's the same number of subunits in both cases, but there's 2 conformations, one with the effector bound and one without the effector bound. And then what Arvind did was to titrate in the effector, and this is sort of looking at the population of the unbound and the population of the bound. And you can see there's this incredibly cooperative transition where we don't really see intermediates. And the cooperativity of this, for those who are familiar with sort of classic protein biophysics, is the Hill coefficient is nearly 4. So this is about as cooperative as the system can be. So we can start designing protein assemblies that have some of the sort of higher order features of native proteins. Okay, so that was all a little bit abstract. Here's kind of a neat application of these ideas. So there's a general problem in biochemistry is, let's say you have an interaction and you want to replace say this partner with something else. You generally will have to wait for the complex to come off. So if you have if you have A, B going, and then you add C, and you want to get AC plus B, even if C binds more tightly than B, the rate limiting step can be dissociation of A from B. And this can be, if it's a high affinity interaction, this can take a long time. So Adam Broerman came up with this sort of allosteric mechanism to accelerate. And the idea is you have the B protein has a binding site for an effector, which is actually somewhat disordered. And so when this binds, it folds upon binding, and that leads to a conformational change in the effector that leads to dissociation of the partner. And this works really well. I have more examples here. So these, in each case, I'm showing, well, these are different of these switching systems. And so this is looking at this is showing you the increase in the off rate as you add in the effector. And you can see it's very high. We get a fold acceleration of nearly 2,000-fold. And Adam can change the extent to which of strain in the system in the different states and tune the amount of rate enhancement. And crystal structures of these designs are really neat in showing how that strain, how the conformational change plays out. But the kind of the biological experiment that this enables is kind of neat. So cytokines are really important signaling molecules, and they bind to their receptors and they cause dimer formation. And it's been thought that the duration of the persistence time of the dimer is really important for downstream signaling, because they have tyrosine kinase domains on their intracellular side, which cross-phosphorylate each other. And so the longer you hold the two together, the less favored phosphorylation sites will be phosphorylated. And so the problem has been that there's no way really to control the lifetime of those complexes. So to probe that, so Adam designed a protein which binds to the IL-2 receptor and has this switching property, so that when an effector is added, you can see what happens. This comes down here and it clashes with this receptor subunit. Oops. That is what that leads to is fast dissociation of the receptor subunit. And you can sort of see this biochemically here. But Adam is collaborating with Jacob Peiler's group, who does this amazing on-cell single molecule fluorescence, where basically they have the two receptor subunits fluorescently labeled, and they can follow they can identify complexes which are pairs of receptors that are co-localized when this compound is added, and then they can follow how fast they come apart when the effector is added. And so that you can see here. You can see within seconds after addition of the effector, the subunits are coming apart, and signaling is increasing. This is when the blue protein is added, it increases to here, and then the effector is added, and the thing starts coming apart. And we're currently trying to apply this concept pretty generally now to make cytokines and other signaling molecules that are only active exactly when you would want them to be active for sort of more precision medicines. I've had some great collaborations with people like Christine's lab on sort of thinking about proteins for plants. And most of what I've talked about was more health related, but we've been Nate Ennis in the group has been very interested in, can we enhance photosynthesis? And so at the heart of photosynthesis is what's called the special pair, two chlorophyll molecules which are exotonically coupled. It's happening within the photosystems, which are really complicated proteins. And what Nate did was to design a small protein which forms a dimer, which holds these two chlorophyll molecules in almost exactly the arrangement they are in the photosystems. So then you can start studying how the geometry of these, how they're located, positioned relative to each other, affect the photophysics. And he's So that's sort of like where charge separation would take place. And in nature, for light harvesting, you have these very complicated light harvesting system. And again, Nate would like to sort of to recapitulate that. Nate's been designing these rings of protein, these protein rings, which have basically a ring of chlorophylls. If you look from the top, it's a little hard to see here. And he can add in other cofactors. And so this would be the light harvesting unit. And we can also assemble these special pair proteins into higher order structures. These are the cubic cages designed by Shunji and Nate that where each edge harbors 2 chlorophyll molecules. And so we're excited both about trying to enhance the efficiency of naturally occurring photosynthesis, particularly the capture of sunlight in wavelength ranges where plant current systems don't do very much, as well as designing completely de novo things for sort of artificial photosynthetic systems. Okay, and then the final thing I want to tell you about is enzyme design. So this is an interesting problem because enzymes in nature catalyze all sorts of chemical reactions. There are a lot of chemical reactions they don't really catalyze, like plastic degradation, where we'd like to have really good catalysts. And so we can do the same kind of thing I described before. If we have an idea of what an ideal active site will look like, we can sort of position side chains around a transition state for the reaction, and then build up a protein around that. And so this illustrates this. This is work from Anna and Sam by taking a really classic reaction, the serine hydrolase reaction, and we're now building up proteins that will harbor a catalytic triad. And this has been really, really interesting because we can now kind of do the opposite of what you see when you read a biochemistry textbook. Usually you see the structure outwards and on the outside, and then you see the details of the active site, and then there's a story about how that catalyzes the reaction. What we can now do is just describe different active sites. So this is sort of an intermediate complexity one where there's an asp his sear, and here's the substrate. And here's a more complicated version where there's there's in addition oxyanion hole residues that provide stabilization for the oxyanion. So we can provide these different geometric descriptions of active sites and then build up proteins that contain these different active sites. And then we can measure activity. So we find with the really simple active sites, we don't really get reaction turnover at all. We do succeed in making the serine nucleophilic, and that's pretty much it. As we add in additional elements, like the aspartate back here, we get turnover, but it's just a single turnover. And the reason why is that we end up getting stuck in an acyl enzyme intermediate. And then as we build up more elements, like the multiple oxyanion hole contacts, we can actually get proteins that carry out multiple catalytic turnovers. And when we solve the structure, when Sam and Anna and Asim Berra solve the structure, what we find is that everything is really exactly in place as designed. So here's the serine, the histine, the aspartate, and the diffusion process has added some more residues that are kind of backing up the aspartate. And here you can see interaction stabilizing the oxyanion. So this kind of enables a new approach to enzymology where you kind of make up sort of ideal active sites, then you build proteins that have them. And we've been doing this for a number of different types of reactions. Here's a similar reaction, but instead of using a catalytic triad, Seth and Donghu are using a metal to activate a water molecule. And the water molecule then it can attack the substrate directly. And it used to be that we tried to do this using the previous generation of methods, we would have to test a lot of designs and only get very small levels of activity. But in testing a relatively small number of designs, Donghu and Seth were very excited to find this one, which had this incredible activity. It's very, very fast, kCat/km of 22,000 per mole per second. We used to be happy if we could get over 100, so this is really a lot faster. And what we found in the process of these enzyme design efforts is that we have some pretty good in silico proxies now that correlate with activity. So we've developed a method called ChemNet that basically you give it some region of a protein, you completely randomize it, and then you denoise it to generate to predict what structure it has. We find that the not very good designs have highly variable substrate positionings, whereas the good designs, like this very active one, has the substrate almost perfectly held in place. And when we look at the catalytic side chains, we also find that those are really locked in place. The way we generate these ensembles is just to carry out the denoising process many times, starting from different random number seeds. So as we develop, it's kind of exciting. As we develop, more sophisticated designs, we're also developing metrics that help evaluate the next generation of designs. So the ones we pick should be even more active. All right, well, that's pretty much what I wanted to tell you about today. I'm really now excited about the applications for protein design. In medicine, I've talked about Neil's work on vaccines. We also are making proteins that bind to the major potentially pandemic viruses. For cancer immunotherapy, we're making proteins that bind very specifically to cancer targets, and as I mentioned, sort of conditionally active cytokines. We're also doing a lot of work now making proteins that bind very specifically to peptide MHCs, sort of like synthetic TCRs for targeting tumor cells. I talked a little bit about drug delivery. On the technology side, I talked about the pores for sensing and protein sequencing, talked a little bit about protein silicon devices. And then on the sustainability side, I'm really excited about this area. Now that we can design enzymes, we can design proteins that break down plastic. We're thinking about sequestering CO2 and with Mary Lidstrom here, methane. I talked a little bit about artificial photosynthesis. So I've had really amazing colleagues who have done all of this work. And I think I've described most of their contributions here. I guess Rosetta fold and RF diffusion, been developed by Rohith. Frank Mink-Young developed the first Rosetta fold version of Rosetta fold. Joe and David and Nate and many others contributed to RF diffusion. Matthias designed the TNF receptor binders I showed you. And the peptide binders were designed by Danny and Anna and Keja. And the nanopore sensor work was done by Samuel and Sagardip. Alexi and Jinwei are just making the designs for silicon. The DNA binding work was done by Cameron and colleagues in the lab. I talked about the delivery stuff, and Joe and Nate did the work on the antibody and TCR design. And the enzyme design work I showed you was by Anna and Sam, and then Seth and Dongyu. I think, I think I mentioned gave credits here. Yeah, I have great colleagues at the Institute for Protein Design. So thanks for your attention and happy to take any questions.